because it's one of the most dependable methods for controlling microbes. So before we even knew what microbes were, we were already using heat, using fire, and boiling things to help make things safer. Because heat works by denaturing proteins, it's going to disrupt membranes and damage nucleic acids. So if you remember our proteins and nucleic acids, they're held together into their three-dimensional structure by hydrogen bonds. So these are really weak bonds that can be broken or disrupted with heat. Also, when we're talking about microbes with heat, we need some really important numbers. So one of our important numbers is the thermal death point. And this is the lowest temperature that kills all microbes in a sample in 10 minutes. We also have the thermal death time, and this is the time it takes to sterilize at a set temperature. I just realized I don't have a bullet point, but you should still recognize what it is, circled over here. This is the decimal reduction timer, the D value. And this is the time needed to kill 90% of the microbes under certain conditions. So you probably don't really care about what the D value is as a microbiology student or future healthcare professional, but food processors do. So food processors use D values to make sure that our canned goods are safe from Clostridium. So they design something called a 12D reduction, and what this says is it makes sure that there's only one spore, if any, remaining before it gets canned and sent out. So basically, it's like taking a population of 1 trillion microbes down to 1. So not that bad. Also, when we're talking about using heat, we have 2 types of heat. We have moist heat and then we have dry heat. So moist heat is basically heat with an assist. So it's more effective than dry heat because water transfers heat more efficiently than air does. So that's why if you stick your hand, don't do this. If you stick your hand into a 350 degree Fahrenheit oven briefly, don't touch the metal, but if you stick it in there, you don't really get burned. But if you stick your hand in boiling water, which is 212 degrees Fahrenheit, you would get burned almost instantly. So that's because moist heat is going to damage those same things, our proteins, our membranes, and it really does make for an excellent disinfectant. It's also going to be involved in sanitation, sterilization, and pasteurization. And the reason why we use a lot of moist heat is because it works fast. So it is a very common tool in both the clinical and the food settings that I just talked about. Boiling. So boiling is really simple. 10 minutes of boiling can kill almost any vegetative bacterial cell, fungi, protozoan, and a lot of viruses. The only downside to boiling is that it won't reliably kill endospores and it won't reliably kill some of the cysts. It is dependent or variable at different elevations. So for example, boiling water in Denver is cooler than boiling water in Miami. Why? Lower air pressure at higher elevations means that water boils at a lower temperature, so it's less lethal to microbes. Okay, autoclaving. So in order to achieve sterilization, we have to use autoclaves. And autoclaves combine both steam, so wet heat, and pressure. So what they do is they usually apply about 15 psi of pressure, and then the boiling point of water rises to 121 degrees Celsius, which is hot enough to kill almost all microbes, including endospores, in about 15 minutes. So really important. However, the steam has to contact every surface. So items are usually wrapped in some sort of porous material. Lids have to be loosened and the air has to get ventilated. So this is why we can't autoclave things that are wrapped in plastic because steam can't get through. So, of course, we don't just hope that the autoclave worked when we run it. We have to use some sort of indicator. So we have used one in lab, and that's an example of a chemical indicator. So autoclave tape. So it changes colors when the conditions are met. Another type of indicator you can have that's not a chemical indicator is a biological indicator. So something like Bacillus endospore strips. If those endospores don't grow after the cycle, you know your equipment is sterile. Okay, pasteurization. So again, we've talked about this. Louis Pasteur, his work on wine spoilage gave us pasteurization. So pasteurization is going to use heat to reduce microbial numbers and kill pathogens without altering taste or nutrition. So here's just some examples for pasteurization of milk. We can have batch pasteurization, high temperature, short time pasteurization, or ultra high temperature pasteurization. So that's what is shown to you here. So batch pasteurization, 63 degrees Celsius for 30 minutes. High temperature is going to be 72 degrees Celsius for 15 seconds. And then we have ultra high temperature pasteurization, which is 135 degrees Celsius for one second. So this can help knock out a lot of pathogens at these various temperatures that we have. So UHT, if we use some sort of ultra high temperature pasteurization, that's great. It's high enough that we only need 1 second to treat it and we don't have any microbes. What the difference between ultra high temperature pasteurization and ultra high temperature sterilization is basically UHT, this one, takes pasteurization one step further. So basically, they're flash heating liquids to 140 degrees Celsius for 1 to 3 seconds, and then then cooling them rapidly. So by doing something like this, we can make products like milk or cream shelf stable for months without refrigeration. So if you've ever purchased like a box milk or a box cream, something like that, that's what it has to undergo. So again, that's why the little coffee creamers, if you use those or seen those at a gas station, why they don't have to be refrigerated. However, the flavor can change slightly over time, but from a microbial standpoint, they are good to go. Okay, dry heat. So sometimes water isn't an option. So if we're talking about things like powders or oils or metal instruments, we want to use dry heat, not moist heat. And it does the same thing. We're still denaturing proteins. We're causing oxidative damage, but we're going to need higher temperatures and longer exposure times in order to kill these microbes. So probably about 160 degrees Celsius for 2 hours instead of like 140 degrees Celsius for a second. You know what I mean? We also have incineration. We do that in lab. This is the ultimate kill step. So when you're